Next up, we have Amanda Eness. She is a Ph.D. student in environmental engineering. Her presentation is on reducing antibiotic resistance in wastewater. Amanda, please welcome to the stage.
Thank you, Karina, for the introduction. As Karina said, my project is focused on reducing antibiotic resistance within the wastewater treatment process. Treatment plants were designed to clean wastewater. The challenge is treatment plants were not specifically designed to clean emerging contaminants. And a big one is antibiotic resistance. And it's a global threat. A recent UN report estimated that by 2050, we could have 10 million deaths a year just from drug-resistant infections. And one of the places where antibiotic resistance can grow is in the wastewater treatment plant. Treatment plants are highly biological environments where bacteria thrive and mingle. And we know that antibiotics and antibiotic resistance genes are present throughout the treatment plant. The goal of my project is to find an efficient and cost-effective way to reduce the spread of antibiotic resistance within the treatment plant, and without adding any extra steps to the treatment process. The wastewater treatment plant is split up into two portions, solids and liquids. From the literature, we know that antibiotic resistance genes are concentrated within the solids treatment process. So I focus on the solids treatment. Solids from the primary and secondary sedimentation tanks settle out and are run through the anaerobic digester. This is what comes out of the digester, dark, sludgy, stuff that does not settle out on its own. Since this is close to the end of the treatment process, it doesn't smell too bad, at least to me. This sludge still has liquid that we need to get out. So utilities add what's called a polymer flocculant. The polymer makes the solids aggregate or clump together like this, resulting in a clear solid-liquid separation. Mechanical dewatering is then used to either squeeze or spin out that final liquid. Biosolids leave the plant, while that liquid is recycled back through the treatment process. The problem is any antibiotic resistance genes still present in that liquid now is going back into the treatment process and mixes with all that new sewage that's coming in. In other words, the treatment process itself could contribute to increasing antibiotic resistance within the treatment plant. My research is to try to reduce this. I work back at that polymer flocculation step, researching which polymers and at what dose is better at reducing antibiotic resistance. Utilities have a lot of choices of polymers. I focus on two types, linear and branched. I then compared dosing of each of them. I wanted to see which polymer and at what dose could achieve an effective reduction of antibiotic resistance genes. But first, I needed to find that optimal dose, which is the amount of polymer needed to get that clearest liquid separated. I found this out by running jar tests. I would set up beakers filled with wastewater, start the mixing paddle, add a dose of polymer flocculant. Right away, that water begins to separate. I would then stop the mixing and allow settling to occur. This process frees up much more liquid that otherwise would not separate. This is a better image of what it looks like. I did this process many times before I could find that optimal dose. Then I took that optimal dose and split it in half. Now I have my two polymer dosages. With that separated liquid, I filtered, extracted the DNA, and quantified specific genes in that liquid. I targeted 3 genes used in antibiotic resistance research. In Taiwan, which is indicative of mobile multi-antibiotic resistance. SOL1 encodes for sulfonamides, which is the first class of antibiotics mass-produced. And BLA-CTX, which encodes for last resort antibiotics. First, I looked at results using branched polymer at those 2 dosages, half and optimal. As you can see, half that polymer dosage achieved up to a 75% reduction of BLA-CTX, which is those last resort antibiotics, which is really important to remove. But half that dosage barely did anything to remove in Taiwan, which is not ideal. Then I looked at optimal dosage. I measured up to a 99.9% reduction of all 3 targeted genes. So with a branched polymer, optimal dosage is kind of needed. Then I looked at linear polymer. At that half dosage, I measured a 97% reduction of lost CTX and a 66% reduction of in Taiwan. Then, at that optimal dosage, I measured again up to a 99.9% reduction of all 3 genes. So, by choosing a linear polymer, utilities could save a little bit money by using a little less polymer, while still achieving that good reduction of antibiotic resistance genes. What we've learned is there are antibiotic resistance genes that, if untreated, are recycled through the treatment process. And we know that treatment plants use polymer flocculants. But by choosing the right polymer structure and optimal dosage, this can be a way to reduce the spread of antibiotic resistance within the treatment plant. Thank you. Good luck.